Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biotechnology conducted in the year 2019. I will be discussing only the biology part, and if you are new to my channel, please do like, share and subscribe. So coming to the first question. Amino acid at its isoelectric point when placed in an electric field exhibits which of these properties? So it exhibits no movement because it is at the isoelectric point. So answer is option C. Then pH of blood is maintained by, this is a repeated question. So that is carbonate and bicarbonate concentration. Not only for integrated MSc PhD this was asked, it was asked for other MSc entrance examinations also of the same university. Then nervous system in vertebrates originates from the following, that is ectoderm. Answer is option A. Then which of the following pairs is incorrect? Amyloplast store starch in potatoes, chloroplast contain ATP synthase complex, chromoplast store pigments in flowers, and etioplasts formed in intense light. Etioplasts are chloroplasts that have not been exposed to light. They are usually found in the flowering plants grown in the dark. So the answer which is incorrect is etioplast formed in intense light. Then which of the following vectors can carry the largest size of DNA insert? It is yeast artificial chromosome or YACS. That is answer D. What can be measured with DNA microarray? So it is the relative mRNA abundance that can be measured by DNA microarray. That is option D. Then the T cell receptor can bind to antigenic peptide only when loaded onto MHC molecule. This is also a repeated question. Then cytotoxic T cells are distinguished from helper T cells by the presence of CD8. This is also a repeated question. Then which of the following isotype of immunoglobulins are both membrane bound and secretory? So it is IgM. Answer is option B. Then next one. Which of the following are professional antigen presenting cell? So B cells. Answer is B cells because classical antigen presenting cells or professional antigen presenting cells include dendritic cells, macrophages, Langerhans cells and B cells. Follicular dendritic cells are specialized types of antigen presenting dendritic cells that are largely restricted to lymphoid follicles. So the answer here is an option A, that is B cells. Then next one. Which of the following is true about blood transfusion reaction, erythroblastosis fetalis and autoimmune hemolytic anemia? They all are type 2 hypersensitive responses. Answer is option A. Linked gene always exhibit. So they have a recombination frequency of less than 50%. Answer is option A. Then which of the following statement is incorrect concerning an X-linked recessive trait in humans? An affected woman almost always has an affected mother. So females, in case of X-linked recessive trait, they get the defective gene either from her mother's defective X gene or if her father has the disorder. So she will gain the X-linked gene either from the mother who is having the defective X gene or if her father is having the X-linked recessive trait disorder. So the answer is option B, that is an affected woman almost always has an affected mother. No, it can be the affected father also. So that is incorrect. Then how is colchicine useful for studying chromosomal mutation? So colchicine, it inhibits microtubule polymerization which stalls the cells in metaphase with condensed chromosomes. So answer is option C. The next one. You are studying a new virus with a DNA genome of 12 kb. It can synthesize DNA at a rate of 400 nucleotides per second. If the virus uses theta replication, how long it will take to replicate its genome? So theta is bidirectional replication. Given 400 nucleotides per second. Since bidirectional, it can add 800 nucleotides per second. That is 400 plus 400. So 800 base pair will be synthesized in 1 second and 1200 1002 so 1 so 12,000 base pair, that's 12 kb, will be in 15 seconds. So answer is option B, 15 seconds. Then what is the function of DNA gyrase? So DNA gyrase, it reduces the torsional strain that builds up ahead of the replication fork as a result of unwinding. Answer is option C. Then next one. Telomerase activity is most likely to be found in which cells in humans? So it is the germ cells that are actively dividing. So it is found in the germ line. Then over time, DNA replaced RNA as the primary carrier of genetic information, and the chemical stability of DNA is believed to be the key reason for this. Which attribute of DNA is the reason behind its chemical stability? So DNA lacks a free hydroxyl group on the 2' carbon atom of its sugar. Answer is option A. Then in the diagram below, which letter indicates the 5' end of the leading strand? So it is the C that indicates the leading strand. So lagging strand is synthesized in fragments, whereas leading strand is synthesized continuously. So the answer is option C. Then which of the following correctly describes the concept of alternative splicing? So multiple proteins products are often produced from single eukaryotic genes. Answer is option. Then the genetic code is said to be degenerate because there are more codons than amino acids. Answer is option A. Then where would the lac repressor be bound in a wild type E. coli cell that is growing in low glucose and high lactose? I is lac repressor gene, ZYA are lac operon structural gene, and P is the lac promoter, and O is the lac operator. So low glucose, high lactose. So E. coli cell will start utilizing lactose. So it needs the lactose metabolizing machinery. Hence repressor would not be bound. Answer will be the repressor would not be bound. Answer is option D. Then antibodies are to western blots as dash are to southern blots. It is DNA for southern blots. Answer is option C. Then dash is the product of sampling errors and chance events that may result in changes in allele frequency. So it is genetic drift. Answer is option C. Which of the following statement about naturally occurring fatty acids is true? So the double bonds found in fatty acids are nearly always in the cis configuration. Saturated fatty acid chains can pack closely together. Unsaturated fatty acid produce flexible fluid arrays because they cannot pack closely together. All of the above. So all of the above is true. Then which of the following lipid is a component of inner mitochondrial membrane? So it is cardiolipin. Answer is option A. Generally, a PCR reaction involves following steps: attachment of primers, denaturation of strand, DNA synthesis, heating. Which is the correct order from starting to the end of the reaction? So first one is heating, that is 4. Then comes denaturation of the strands, 2. Then comes attachment of primers, 1. And then comes DNA synthesis, that is 3. So it is 4, 2, 1 and 3. Then chromatography is the process for identification, purification and separation of components of a mixture on the basis of. So it is on the basis of difference in the affinity for mobile and stationary phase. Answer is option D. Then find out the mismatch of the following. So magnesium nitrate reductase. It is not magnesium, it is molybdenum. So that itself is the mismatch. Answer is option A. Km value is expressed as the substrate concentration at half Vmax. Answer is option B. Then C4, during C4 pathway of photosynthesis, malate is transported from mesophyll cell to bundle sheath cell. Answer is option C. Then the radioactive molecule that can be used in the analysis of protein is, it is S35 methionine. Answer is option C. Then hybridoma technology is being used in, it is used in generating monoclonal antibodies. Answer is option A. Then next one. Which of the following statements about antibiotic resistance in bacteria is not true? So antibiotic resistance cannot be conferred by conjugation, which only affects the fertility of bacteria. So that is not true. Answer is option B. Then which of the following statement describes the Wobble rules correctly? The answer is option D, that is the third base pairing between tRNA and mRNA is relaxed. So answer is option D. Then modification of protein as they pass through Golgi include all of the following except signal sequence removal. The signal sequence is removed co-translationally by the signal peptidase located within the endoplasmic reticulum lumen. Then next one. The marker for an enzyme to go from Golgi to lysosome is, it is phosphomannose. Answer is option C. Then uncouplers of electron transport from ATP synthesis such as dinitrophenol act by inhibiting the proton gradient. Answer is option. Then which statement is not true of gap junction? They allow the passage of proteins between cells. So molecules like inorganic salt, sugars, amino acid, nucleotides and vitamins, but not large molecules such as proteins or nucleic acids are allowed by the gap junction. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.